Hello, I'm happy to be here at the virtual RPPW presenting new research on the genetic architecture of rhythm and language traits. So we're already learning a lot about the biology of rhythm from neuroscience imaging methods, as presented in Anna Kasdan's talk in this session. So what can genomics approaches tell us about the biology of rhythm? Well, as we get a foothold on the underlying neurobiology of individual differences through genetics, we can ask, what are the genetic contributions to the neural architecture of beat synchronization? In other words, how do genes influence the brain network's underlying rhythm ability? We can also use genetics to ask questions about the biology driving individual differences and correlations between rhythm and language. Perhaps some of the genes that influence rhythm could also influence language skills. The method that we are talking about today is genome-wide-association studies. So Qtl-Gwas gives us associations between a phenotype and genetic alleles at strategically measured genetic markers across the genome. This tells you what genes influence a trait. Let's quickly highlight a key concept of Qtl-Gwas. Qtl-Gwas uses common genetic variation to show us small effects that are widespread throughout the population. That's different than detecting rare variants or mutations that would cause large effects at a single locus. Now, rhythm is a complex trait with a variety of individual differences in the population. So Qtl-Gwas could tell us more about these individual differences and lead us to the underlying biology. So for today, I just want you to note that the Qtl-Gwas output will also allow us to test for shared genetics with other traits. For our study, we were fortunate to partner with 23andMe. They have data on millions of people who are already genotyped and have also consented to research. In our study, over 600,000 people answered the self-report question, can you clap in time with a musical beat? So this item became the phenotype for the Qtl-Gwas study. Our preprint of the Rhythm Qtl-Gwas study is on BioRxiv, and there'll be another revision of it posted later this summer. I'm going to tell you now about a couple of highlights of this first study, and then I'll describe how we're using the data to look at rhythm and language. As I'm sure this audience can appreciate, it was really important to validate the self-report item. We did this in a number of ways using rhythm perception and production measures in 2 phenotype validation experiments. Those data are described in our preprint. Just briefly, I'll point out the results of the experiment where we asked people to tap in time to the beat of musical excerpts. And for this, we used a new audio algorithm developed by Nori Jacoby's group. The results here are plotted with beat tapping accuracy as the standard deviation of the asynchrony. So lower values mean better accuracy, closer to the beat. So you can see that people who respond yes to the self-report are generally much more accurate at tapping to the beat of music than the no responders. From these studies, we were able to conclude that the self-reported beat synchronization item is a good proxy for measured rhythm phenotypes. Okay, now back to the Qtl-Gwas portion of the study. So here we have the Manhattan plot of the Qtl-Gwas study in 606,000 23andMe participants. So this shows us genetic associations with answering yes versus no to the clap beat self-report question. In other words, these are the strength of the association of alleles with beat synchronization. And here, statistical significance is on the y-axis with this gray horizontal line as the threshold after multiple test correction, and the dots are genetic markers lined up on the x-axis by chromosome. We found that 67 independent genetic loci are associated with beat synchronization. So beat synchronization is highly polygenic. And there's so much signal here in this Qtl-Gwas. So we mapped these markers onto their genes as a way to reduce the dimensions of the data. And then we asked, what are the properties of all of these genes that are associated with beat synchronization? So one way we can look and explore this question is using gene property analysis on our Qtl-Gwas results using GTEx predictor data. GTEx is a reference set where gene expression levels are characterized in 54 tissues in the body. Our analysis will let us see tissue-specific gene expression in relation to our beat trait, but based only on the Qtl-Gwas data. So So this means that we can get a sense of the tissue specificity of gene expression in brain tissues and in other tissues linked to our beat synchronization phenotype without directly assaying those tissues in the participants. And that's one of the many ways that we can triangulate between DNA, gene expression, the brain, and rhythm. The results of this analysis show that the genetic architecture of beat synchronization is highly enriched not only for gene expression and brain tissue overall, as expected, but in particular for gene expression in cerebellum, basal ganglia, and cortex. So these results really align nicely with cognitive neuroscience evidence of the neural substrates of beat synchronization and perception. So we're really interested in whether musicality and language traits share genetic architecture more generally, and then specifically whether beat synchronization shares genetic architecture with spoken language and reading traits. And that's a prediction that we made based on phenotypic associations in the literature, including some work from my lab as well as many, many other studies. So to explore the concept of shared genetic architecture as we will apply it today, I want to briefly review the concept of pleiotropy in complex traits. So pleiotropy is when a single genetic locus can affect two independent traits. Biology research has shown for many years that cross-trait phenotypic correlations reflect underlying genetic correlations due to pleiotropy. A huge advance in genomic methods in 2015 gave us a method for testing cross-trait pleiotropy in data from separate samples, and then subsequent studies have used this method to show pleiotropy among many cognitive and health traits. So this method opens up all kinds of doors to integrate results across separate data sets, as I'm sure you can start to imagine. So the way that we're going to look at whether rhythm and language are genetically correlated is to access language genetic summary statistics. There's a big GWAS project underway with the Genlang Quantitative Trait Working Group led by Simon Fisher and Elsa Eising. And this is a massive collaboration where they are meta-analyzing data from 21 different cohorts that have language scores and genomic data. So in the interest of time, I'll just highlight the 5 different language constructs in their study that we will also be using in our study, and the ends for each trait here in this row, and examples of the specific language assessment. So here, this is not self-report. These were done by language testing. Using that data, we've been able to compute genetic correlations between the beat synchronization GWAS and those language-related GWASs. We were very excited to see genetic correlations between beat synchronization and each of these reading-related traits, word reading, non-word reading, spelling, and phoneme awareness, as well as non-word repetition, which is a good proxy for spoken language and even spoken grammar skills. So these results really align nicely with findings from the literature of behavioral correlations between rhythm and reading and between rhythm and spoken language skills. So what's next? Well, genetic correlations indicate overall shared genetic architecture, but they don't tell us which loci overlap or what those genes do. So we'll need to dig in with other methods to know which genes are shared and what their functions are. Now remember, language skills exist on a continuum, so these studies will not only be interesting from a basic science perspective, but also focusing on the risk aspect. It becomes possible to think of atypical rhythm in terms of identifying risk factors for speech and language disorders. And we discuss this prospect in our Atypical Rhythm Risk Hypothesis paper, where we talk about elevated presence of atypical rhythm skills in speech and language disorders such as dyslexia, stuttering, and developmental language disorder. We propose that these associations could be a reflection of genetic pleiotropy between rhythm and speech-language development. So the genetic correlation results that I showed you today are a first form of support for that prediction. So let's summarize what we've discussed today. Beat synchronization is highly polygenic. Many genes are involved in this trait. The genetic architecture of beat synchronization is enriched for gene expression in brain tissue, and in particular for cerebellum, basal ganglia, and cortex. Preliminary genetic correlation results suggest shared genetic architecture between beat synchronization and language and reading traits. This may have implications for risk detection in speech and language disorders. And if you're interested in hearing about groundwork for eventual genetic studies of speech rhythm, check out the poster by my postdoc, Srishti Nayak, in session 4 on Friday, validation of a novel speech rhythm perception test, convergence with musical rhythm and language and reading difficulties. I am extremely grateful to my research team, my collaborators, our funders, and the research participants for making this work possible. And thank you for your attention.